And of course, we want you to be engaged. So please drop your questions and your comments in the chat. But we do ask you to be a good digital citizen. Let's keep the questions and comments relevant and things positive and respectful. To get that chat going, I want to know if you have any scientists in your family or if you are friends with scientists. We're going to be joined with not one, not two, not three, but 4 excellent students from NC Central University. And so they're going to tell us all about the cool research they're doing there. And so we just want to know if you know any scientists. So now that we're all familiar with Zoom, I'm going to go ahead and turn it over to Dr. Julie Horvath. She is the head of genomics and microbiology at the North Carolina Museum of Natural Sciences, but she's also a professor at NC Central University. Take it away, Julie.
Thanks, Greg. Well, thanks for joining us, everyone. As Greg mentioned, my name is Dr. Julie Horvath, and although I am the head of the Genomics and Microbiology Research Lab at the museum, I'm also jointly appointed at North Carolina Central. And so I'm really excited that I have some of my students here today. So I teach in the springtime 2 graduate seminar courses to both master's and PhD level students. And these students get experience communicating their science to a variety of different audiences. And so today you get to hear about some of their exciting research. And I'm really excited to have them here because they have really great projects. And even though they're early in their research careers, they have a good handle on some of their topics. So you're going to hear about some really relevant research that's probably relevant to a lot of you in your daily lives. So first, we're going to start off with Asia Thomas talking about zebrafish research. Then Donia Kased will talk about cancer in minority populations. Nadine Thomas is going to continue talking about cancer, but from a different perspective. So she's going to focus on the use of natural products to find new treatments for cancers. And then Jeffrey Shipman is going to end us out today talking about vaping and COVID-19. So I hope you learn a lot. I hope you ask some questions after their talks and hope everyone has fun. And thank you all for joining us. So with that, I want to hand it over to Asia Thomas. So Asia, if you want to start sharing your screen, and you can unmute and take it away and tell us about your research.
Hello, everybody. My name is Asia Thomas. I'm currently a first-year graduate student at North Carolina Central University in the Biological and Biomedical Sciences program. I currently work in the lab of Dr. Lee doing research on zebrafish. Upon graduation, I would like to go get my MD and focus on becoming a medical examiner. So today I'm going to talk to you guys about zebrafish and how they are possibly the future of research. Give me 1 second, it is messing up. All right, to start you guys off, I'm going to give you some terms that you may not know about. A transgenic organism is a genetically modified organism with an altered genome. A fluorophore is a fluorescent chemical compound that re-emits light. And intestinal metaplasia, also known as IM, which is a condition where stomach lining cells resemble intestinal cells and it can become cancerous. A little bit of background information about zebrafish. They are one of the animals used to generate transgenic organisms. They are used because they're relatively low cost, easy to maintain, and the embryos develop relatively quickly. There is also a high yield of eggs in the lab that I work in. We currently collect eggs. We collect eggs on Wednesdays and Fridays, and we normally have about 200 eggs per tank that we have for the day. And the eggs are transparent, which makes it very easy to observe their development. And if you look on your left-hand side, you can see where my pointer is, how the zebra eggs are transparent and you can really obviously see through them. These are the cells and they divide as time goes on. All right, so to create a transgenic zebrafish, it is relatively easy. What you have to do is first isolate the gene of interest in which you are studying and tag it with the fluorophore, as I mentioned earlier. You would then collect the zebra eggs that have been laid and clean them in purified water. Once you have done that, you will observe them under the microscope, getting rid of the bad eggs and injecting the gene into the embryo. Over time, you care for those eggs as if they're children because you want to make sure you have viable offspring so that way you can create a transgenic line of zebrafish. If the gene isn't incorporated into the genome, then under a fluorescent microscope, you can look for glowing areas. They could be green, red, is different colors that you can also use. As you see on the right-hand side, you can see a zebrafish that has a fluorophore, and the fluorophore is green, causing it to glow green. All right, so I'm just going to give you a little bit of background information about genes to know about. You should first know about the CDX2 gene, which is an encoded protein and is a regulator of intestine-specific genes involved in differentiation and cell growth. And then you also have CDX1B, which stops DNA binding transcription factor activity and sequence-specific DNA binding activity involved in the digestive tract of zebrafish. And I didn't mention, but the CDX2 gene is found in humans. Now, a study conducted in the lab of Dr. Lee at North Carolina Central, he wanted to study whether the gene CDX2 had a role in developing intestinal metaplasia in humans. So he did that using the zebrafish. He used the zebrafish with the gene CDX1, which is the equivalent of the CDX2 gene, and overexpressed it in genes to see if it would have an effect on the stomach lining cells. A transgenic line of zebrafish was created with the CDX1B gene overexpressed, and there was a wild type line of fish that didn't have the CDX1B gene overexpressed within it. It was found within that transgenic line of fish that overexpression of the CDX1B gene within the stomach cells failed to induce intestinal metaplasia. However, it did change the morphology of those cells. So one of the methods used was A and C2 hybridization, which is where you have single-stranded DNA or RNA probe is allowed to form complementary base pairs with DNA or RNA present in a tissue sample. And then you have B, which is immunohistochemical staining, and that is when you look at the morphology of the cells so you can see how they are growing. And then we also have 3, which is real-time PCR, which is a technique that combines reverse transcription of RNA and DNA, and it amplifies the specific DNA targets using PCR. And real-time PCR is what they use today with detecting the COVID virus. It detects the presence of a specific genetic material within the pathogen. In conclusion, it was said that although intestinal metaplasia was not induced within these fish, it did show us that we can study how the gene does affect the stomach lining cells, which can be useful in the future. For example, with transgenic animals, you are able to knock out genes to how you see fit and study that gene. This has helped us in the field of cancer research and has given a rise to CRISPR and Cas9, which is a gene editing software. Soon, scientists hope to be able to alter the human genome to possibly stop diseases such as sickle cell when there is a change in amino acid codes that can cause an sickle cell disease to happen, which is when you have C-shaped blood cells. And you may ask, why is this relevant to today's information? Without transgenic organisms such as zebrafish, we wouldn't know many things about science because it is unethical to work on humans depending on the work that is being done. With transgenic animals, as I said before, you can alter the genome how you see fit. So you can knock in genes, knock out genes, things of that nature. And transgenic organisms have also gave us the ability to make insulin, which is when bacterial cells can be genetically modified to have the gene needed for insulin production. In the lab that I currently work in, we're looking at the EDAR gene, which is the ectodysplasion A receptor gene, and it plays a role in the development of ectodermal tissues such as the skin. So if you look over here on your left-hand side, you can see how when there's a mutation in the EDAR gene, it causes mutations within humans. Like you can see how the teeth are not forming correctly, the gums aren't, the skin, the ears, and things of that nature. So currently we're working to create a transgenic line of fish to see if the fish are able to survive. In humans, if this gene is mutated, then it can cause a person to have hypohidrotic ectodysplasia, which is a genetic skin disease. Does anyone have any questions?
Asia, that's fascinating stuff. You know, in your title slide, you said that this fish is the future. I'm wondering more about the origins of how this particular species was discovered. Like, how did researchers know to use the zebrafish to do these types of studies?
Even though they are distant, like relatives, I guess, to humans, it has shown that it would be a good model organism, not because of some of the homologs that they share and how some of the genes are similar, even though they're not named the same. But this research is just, I want to say, about 40 to 50 years old, so it's relatively new.
Interesting. Well, thank you. I'm not seeing any questions in the chat right now. This is a reminder to our audience that if you have questions for any of our presenters, you can drop them in the chat and we'll ask those after they go, or we'll save some time at the very end as well. And as I was saying that, we got one from Paula. So Paula wants to know, where do the genes that you knock in come from?
So when you knock in and knock out genes, you're trying to basically get a, how do I explain? You get in like a 00 phenotype, like there's no phenotype that is going to be expressed, and you want to put in the gene that you're possibly looking for to experiment with that.
Great, thanks. All right, thanks, Asia. Let's move along. We have Donia up next. And Donia, I had a request from our transcriber that if y'all can just slow down a little bit so that they have time to transcribe the captions accurately for those folks watching at home, please. Donia, go ahead and take it away. Donia, you're still muted.
Good afternoon. My name is Donia Kassid. I'm a PhD candidate in North Carolina Central University, and my interest is studying the diseases, understanding it, and studying the components that interact or cause the disease. And my dream is to become a doctor that understands the diseases and studies it as a support or aid to find treatments. Today, I will be talking about my project, The Battle of Science Against Cancers, Mutations in Minority Populations. They say that a picture says 1,000 words. Over here in this picture, every single person looks different, and this is because of genetic variation. We all have similar DNA, but the difference is a few variations. So if we look at the sentence, the girl and the cat ran up the hill. The sentence has a meaning to it. In the second and third sentence, is there a difference? You'll probably be seeing the difference in the second and the third sentence. You can answer in the chat box, yes or no.
I'm seeing a lot of yeses.
Okay, so a majority of us said yes. The first sentence was the girl and the cat ran up the hill. When we change the word and we take the letter A and change it to an E, it's the girl and the end. Even further, if we change the girl and the bat, we change the C of the cat into a bat, the meaning of the sentence changed. So the same thing with DNA. If we have a DNA sequence where the DNA produces a protein, this DNA sequence also has letters. If the letters changes, instead of the meaning of the sentence changes, it will produce a different or a protein that will be a variant. This is called mutations. Mutations has letters, the gene has letters. The GAA changed to the GAC, that's in red. So the protein has a spot on it that's different, it's a variant. It may affect its function or even structure. In the United States, African Americans have higher cases of patients with metastasis, which is cancer or cells that move, tumor cells that move throughout the rest of the body. This causes African Americans to have 33% higher chances of dying due to cancer. This may be impacted due to socioeconomic statuses, behavioral factors, Even further, it can be because not everybody lives close by a hospital. In combination with our study suggesting that STIC1 mutation may increase the risk of metastasis, which is cancer spreading out throughout the body. STIC1 protein is STIC1 is called serine thymine tyrosine kinase 1 protein. This is a protein, and in our study, we found mutations that were found in higher frequencies in African Americans. These mutations were found in many cancers, such as prostate cancers, liver cancer, breast cancer. And what is the function of this protein? Well, the function of this protein, it has a role in cell development. Since it has a role in the cell development, when it's mutated, this is a major issue because cell development also covers the cell growth, the cell expansion, also cell survival. What if the cell can survive a lot or can expand or move throughout the body? This is a major issue. We found 3 STIC1 protein mutations in African Americans that were found in high frequencies, so we decided to study these mutations. How does STIC1 actually impact the process for it to cause metastasis? Well, STIC1, it blocks the glue. If we have 2 pieces of paper and we want to glue these 2 pieces of paper together, we need a certain amount of glue. Okay, if the amount of glue is less, the paper won't be sticking together as much. Same thing with cells. Cells also have a glue substance, which is called E-cadherin. E-cadherin is the glue. STIC1 protein that we are studying blocks the production of the glue. So we don't have enough glue. So what will happen? The cells will start being loose and moving throughout the body, which is called, again, metastasis. So our experiment first was to obtain the 3 different STIC1 mutations. Then after that, we did a process to find its size, which is called the SDS page. We even moved forward to measure the activity of these proteins. Are the mutations causing it to be more active, less active? What are the rates? The 3 mutations, when we studied the rates of them, we found that the rates were variant. Each mutation in this table, 1, 2, and 3, had different rates of reactions. The rates of reactions that we studied was the rate in ATP, which is energy. The energy has a phosphate group. STIC1 is capable of removing 1 phosphate. We were studying how quick is this phosphate being removed, and we found that the 3 had different rates. Next, we did an SDS page, which studies the size of the protein. When in the SDS page, we have ladder. The ladder is just like a ruler. If I had a ruler and I put it next to a piece of paper, the ruler would measure the piece of paper. The number that's horizontal from this ruler will give us the size of the paper. Same thing with the ladder. The ladder is just like our ruler. The ruler over here, we will compare to the mutations, which are the proteins. We had 3 proteins, which are the 3 lines appearing here. These 3 lines, comparing to our ladder, which is our ruler, is between 100 kD to 70 kD. kD is the unit. So we can say here that it is about 80 kD. All of them are about the same size. But as we said previously, they have different rates of reaction. In conclusion, different mutations within STIC1 can be found in different ethnic group populations. And this can be impacting their cancer outcomes and also impacting the treatments that are being given to them. The 3 mutations were found at high frequency in African Americans in our study, although there may be other populations that also have different variants of these mutations as well. So the future directions is in our studies to give a further clarity to the high rates of cancer patients within African Americans. And also, we are hoping to try to express these mutations or place these proteins in cell culture. Cell culture is a plate that has cells, and we will be expressing these mutations in it. What we may observe is that once we express the protein inside of these cells, we can see cell growth, uncontrolled cell growth. We can also see survival. It's still surviving for a while, more than the normal treatment. And this will show us if these mutations were actually in humans, will they actually also have the same effect as they are in plates? This will need also clinical trials. later in future. Thus, this project may support future studies informing effective treatments based on the STIC1 mutations found in patients. So based on our studies, we're saying that in the future, if we find that STIC1 mutation, there is a mutation, it's reacting certain ways, then we may, or other scientists may also use this research to find an effective treatment that targets this STIC1 mutation to stop the uncontrolled growth of cancer or metastasis, therefore decreasing the death rates due to cancer. I would like to thank Dr. Nathan Weimer for his continuous support and teaching me in the lab different techniques. I would also like to thank my mother, family, and father for their continuous support and motivating me to go after my goals. Thank you. Does anyone have any questions?
Thanks, Donia. That was great. I'm wondering, can you provide an example how this kind of process of identifying a mutation has been used to maybe treat other cancers? Like, has this been proven effective before? Can you speak to that?
It hasn't been proven effective before. It's still under study. We're so far studying the protein structure and its function. We haven't yet expressed it in cell cultures or in cells. So this is our further studies. I was saying that if it is affecting the cell growth and it's causing uncontrolled cell growth as in cancer, then we may use it or other scientists may use it to find treatments to target this specific gene that's producing the protein.
That's great. So your findings hopefully will be passed off to somebody else who can come up with those solutions to effectively treat cancer. It's kind of a team effort solving these big problems, isn't it?
Yes, of course. I believe that as a team, we can always solve anything.
So I do have one question from the chat. Marianne is asking, is this gene being looked at in patients now? So
far in our study, we're working from the point of view of in the lab. We haven't looked at it in patients, but there was a previous study in the WHO finding that these 3 mutations that we are targeting was found at high frequency in African Americans. We got this data from the WHO.
Great, thanks. And as a reminder, if you have more questions, we can get to those at the end, but small round of applause for Donia. And we're gonna pass it off to Nadine.
Good day, my name is Nadine Thomas, and I am a PhD candidate at North Carolina Central University, and my career goal is to become a research physician. I'm currently a novelist, and I am in the biochemistry and chemistry department. My goal is to use natural products to treat cancer, and my topic or my presentation today is that small molecules are still hot. And so before I start my presentation, I would like to ask you 2 questions. And so the first question is, why do you think that people are so scared of chemotherapy? And my second question is that in the early 1700s, do you think that humans were affected by cancer as we are today? I'll give you some time so that you can respond in the chat room, but you just need to respond with a letter response. You don't need to write out the full options because that will take time. So I'll give you some time for you to respond.
I'm seeing a lot of C's for the first one. Also several E's for question number 2.
Okay.
The general consensus looks like it's C and E.
Okay, so I'll continue then, but I'm really impressed or intrigued by your answer that a lot of people are speaking about the harsh side effects of chemotherapy and also about there were cases, but it weren't diagnosis then. I did ask this question because I do know that currently we do have a lot of questions about chemotherapy and we do have a lot of questions about cancer in general, like is there a treatment for it? Will we ever get to a stage where this will be controlled? And so this is very important or interesting for us. And so my project for today, or my project will be about the CLUSIACEAE family of plants. And this is because it is they have produced small molecules called PRENYLATED benzophenones. And why are they important is because they help us in treating cancer, and they do so by interfering with enzyme pathways. And one such enzyme is your HDAC11. And so this is important, and I will explain HDAC11 in a second. But first, I wanted you to view or take a look at these lovely plants, because these are all from the CLUSIACEAE family, which I just mentioned is very important. Now, HDAC11, I just mentioned, it regulates our immune cells. It's a protein in our body, and specifically, it's an enzyme. It also inhibits cytokine IL-10. Now, what are cytokines? Cytokines are chemicals that are produced in our body by our immune cells. As I show in the picture below, your lymphocytes, your macrophage, granulocytes, your immune cells, they produce these cytokines. Now, IL-10 is just one example of cytokines. So why are these chemicals produced? Because there might be cells in your body that might be going through a viral infection, a bacterial infection, different kinds of triggers, and your body is trying to normalize things. So it sends these chemicals out just so things can be regulated. And so this is to show you that IL-10 is important. All your cytokines are important. And so HDAC11 that regulates it is also important. And so HDAC11, this enzyme is found in your brain, your testes, and your immune cells. So again, I put some picture of different types of your immune cells because this is where you can find your HDAC11. And then an important aspect also is that this HDAC11 enzyme, it has been increased in various cancers. And this is where we have a problem. Why? And that's the aim or what we want to deal with, because this increase in HDAC11 causes IL-10 dysfunction, and that IL-10 dysfunction leads to cancer. What can we do about this? From these plants, we can get the PRENYLATED benzophenones. Why would we do this? It's because we are trying to block that HDAC11 increase. When it's blocked, we'll stop the IL-10 dysfunction, and that would stop or treat or prevent the cancer. And so this is why this is exactly what we did in the lab. And so this is a method that what we have done already. And so the first thing is that we got the plant. This plant is specifically Clusia CUNEATA, and I was very excited to use this plant, and that's because it's coming all the way from Guyana, and it's a relatively new species from the family of plants. And so when you use new species in research, then you can get new results, find new molecules, and contribute something great to science. And so just like how you'd make your callaloo juice, we blend it in your blender. Then we did 3 extractions, and so that was hexane, ethyl acetate, and methanol sequentially. And then after that, we did a second extraction. You see it's more liquefied. And then after that, it was filtered. And then after that, we had a product. And the product is important to us because it was left for a few days so that it could get solidified, because of after evaporation, it's solidified. But this product is what would have our PRENYLATED benzophenone, this compound that would inhibit the HDAC and would be used to help us with the cancer. And so that's why it's important. Now, these results or these molecular weights were collected from previous results, and it's important with us because if you notice in my table, neuromosome, triazepine, ibuprofen, and garcinol, they're all previous PRENYLATED benzophenones, but their molecular weights are approximately between 500 and 600 grams per mole. And so first, your molecular weight is basically the size of the molecule, and this is used to identify them. And so this is what we use also in our experiment. And why is this like important is because first, I also want to point out that there are many ways that you could use to identify that we indeed have prenylated benzophenones. Because of time, I'll only mention or explain this one, but there are several ways that we use in the laboratory. This is just one example. But why is it important? Because this is used to identify the size or the mass of these molecules. And so if it's too small or too big, then we'd know that it's not prenylated benzophenone. So if you notice the number I circled, 515, 516, 533, 545, those are the numbers we're interested in because those are the prenylated benzophenones in the samples that we want, the molecules that we want. And this was done by the mass spec. And the mass spec is the instrument that is used to measure it because it does so from a mass to charge ratio, and it measures or it takes the mass of the molecule, and that's how it identifies it. So in conclusion, sequential hexane, ethyl acetate, and methanol extraction were performed on the CLUSIACEAE CUNEATA plant, and prenylated benzophenones were produced. And now for the future direction, this is also a very exciting part of the project. And this is because this is a part that we're doing right now and it's very important. And so the first is the substrate and HDAC11. And so the substrate is lysine because in our body, naturally, HDAC11 works on lysine, a lysine-containing compound. And what we want to get in the laboratory is a yellow product. And so when you mix the lysine and the HDAC after the series of reaction, it should change the compound from white substance to yellow product. And then we know that the HDAC11 worked. But after, we are gonna mix the same substrate with the same HDAC11, and then we are gonna add our inhibitor or prenylated benzophenones, and we should not get that yellow product. It should stay white. And that would mean what we hypothesize that this prenylated benzophenone can actually inhibit the HDAC11, it actually works. And this would be important for us because we can use it for future studies and we can continue with our research because this will be very great for us. I'll leave time for further questions, but first, I really want to thank NCCU for allowing me to be a part of this great PhD program. I want to thank my fantastic advisors, Dr. Christian and Dr. Weimer, for allowing me to join their lab and doing such fantastic research. I want to thank my grad seminar professor, Dr. Harvard, for allowing me to present to you guys. And I want to thank you, audience, for listening to me. Now, thank you all, and any questions?
Nadine, that was fascinating. You mentioned that the Clusia CUNEATA plant was new. Do you mean it was like recently discovered as it's new to science, or it's just now being used in this sort of biomedical cancer research?
So, I mean, but like it's just recently been discovered. And so, and even when I researched like it myself, I wasn't able to find any like previous research done on Clusia CUNEATA itself, even though other research was done on other CLUSIACEAE plants. So yes.
Yeah, that must be really exciting for your lab.
Yes, it is actually exciting. So yes.
Yeah, and Carrie says, what a compelling reason to protect nature, right? The natural world holds possibly cures for cancer and all these other ailments out there. Carrie was also wondering, how did you decide to use this plant for your research then?
Okay, so for the CLUSIACEAE family itself, it has a whole like wide group of plants that other researchers have used. And so it, as like previous papers have shown that there are like prenylated benzophenones from other plants, but the fact that it's a family, it does indicate that this plant specifically can have something. Since it's family and it's not being researched, then it makes sense to try something new with a different plant from the same family. Also at the same time, where other researchers focus on the other HDACs. As you know, their HDACs have 4 classes, and HDAC11 is like, it's by itself, and not many persons was able to find stuff on HDAC11. And so my hope personally is that since it's a new plant, like a new species, and since HDAC11 isn't studied as much, then maybe we can find something on HDAC11 specifically. So yeah.
Great. Okay, and then we have a couple of questions from Eduardo. First one is, do non-prenylated how do you say that?
Non-prenylated benzophenones.
Yes, induce cancers as well, or only the prenylated benzophenones?
You mean like help with cancer? So far I'm sure about the prenylated benzophenones because of the focus of my research. I think like other like benzophenones, like in general, like it does help, but on the compounds that I've been focusing on or I've been working on, I think like they provide a better help, if that makes sense. Like they help more, or so more papers have been focused on specifically prenylated benzophenones, and it's more based on their molecular weight and their structure, like how their structure is, and that's what make them like so special or so different from the other compounds.
And then Eduardo's second question, are prenylated benzophenones considered octogens in current research?
Are considered? I didn't hear you.
Oncogens? Sorry, I'm not familiar with that term.
I still didn't hear what you said. I'm sorry.
It's oncogenes, O-N-C-O.
Oh, oncogenes, if they're considered oncogenes. So far, I wasn't aware of that. Like, I didn't read it like it was considered oncogenes, and there are a lot of papers that it's still being studied, like it's a relatively new study. And like I said, like while some HDOC, they were positive that it worked on it, some papers are still unsure. And so I think because of that, it wasn't like, I didn't see any paper that definitely said that this prenylated benzophenone is a oncogene going forward.
Great job, everybody. Yeah, lots of positive comments coming through the chat. Nadine, thank you for all that wonderful information and for also teaching me a whole bunch of new words.
Thank you. Also.
We're going to go ahead and pass it off to our final presenter, Jeffrey. Jeffrey, just make sure you unmute yourself.
You guys can see my PowerPoint, right?
Yes, it's not in presenter mode though.
Not yet. Okay. I got you.
Okay. Jeffrey, we're seeing your notes.
Oh, wow. Okay, give me a second. Really strange. There you go. You guys can see this now, right?
Looks perfect.
Okay, thank you. Thank you. Hello, everyone. My name is Jeffrey Shipman. I'm a first-year graduate student in NCCU's Integrated Applied Sciences PhD program, and I currently work in Dr. Vijay Silvaraman's lab. And our research focus is looking at how different factors such as vaping or alcohol can result in lung damage or lead to more severe kind of like lung and respiratory diseases. So coronavirus or COVID-19 is this virus that affects our lungs and leads us to develop like pneumonia. But kind of the factors that lead to the more severe cases of coronavirus aren't better known. But what you can imagine is that things that affect the lungs because we developed this pneumonia would affect the severity of COVID-19. So smoking affects the lungs, and 34 million U.S. adults smoke. And if you smoke in the U.S., this typically leads you to be 1.4 times more susceptible to severe cases of COVID-19. Now, vaping, as you can imagine, vaping interacts with the lungs. 8.1 million people use electronic cigarettes or vaping, but little is known about vaping's effects really on the lungs, as it was originally cited as a safer alternative to smoking, but recently studies have begun to look at vaping's effects. So we want to study what role vaping plays in relation to the severity of COVID-19 or coronavirus. And in order to do that, we use a mouse model. Now, we utilize this mouse model to look at this disease because mice are very similar to humans. obviously we don't look like mice, but there's different biological processes that mice have that are very similar to our processes. So we can look at the lungs in mice to see what kind of reaction you would have in lungs in humans. So in order to do that, we use this mouse coronavirus model, MHV. And MHV in previous papers have shown to kind of mimic that SARS or COVID-like pneumonia that we develop in our lungs as humans. So we have a very clear objective for our research, and as we want to see how vape mice respond to coronavirus, pulmonary challenge, or lung challenge. So in order to do that, we have these groups of mice, and we have control groups, and then we have these groups that were vaped using a mint joule mechanism. So of one of these groups, we also co-expose these groups MHV, which kind of simulates that SARS-like pneumonia infection. And we investigate these three factors to see what happens after this. So we look at the lungs and we look at wet-dry ratio, because wet-dry ratio is used to study lung injury, acute lung injury. We look at inflammation by looking at these cytokines, which Nadine mentioned are these chemicals that are produced by the body to mediate inflammation. And we want to look at the viability of these mice that were vaped and co-exposed to this virus. So what we'll find first is that vaping damages these mice lungs. So on the left, you can see these pictures of these mice lungs. Now, the control and the vaped PGVG, which is the vehicle control. So we use these two controls to compare to what would happen after we vaped them with the mint joule. And you can see that in this vaped mint, that these lungs are like darker and they're smaller and shriveled up. So we use wet-dry ratio to see what kind of lung damage was done here. And you can see on this graph on the right that when looking at this, this vape and this MHV, when exposed to these lungs, that this lung wet-to-dry ratio begins to increase. And this increase in wet-to-dry ratio shows that lung damage is beginning to happen. So next we look at inflammation. Now, IL-6 is a cytokine that is typically viewed to look at inflammation. So what happens is if we see this increase in this IL-6 cytokine, there's an increase in inflammation in the lungs. So when looking at the control in the vape control, the PGVG or the vehicle control, you can see there's this increase of IL-6 in the vaped mint group, which shows this increase in inflammation. So next, we'll look at the viability of these mice and like under vaping and coronavirus. And what we'll see is that there's a decrease in mouse viability in this co-exposed group. Now, vaping alone didn't lead to any major decrease in viability, but you can see this decrease begin to happen with MHV, and then the co-exposure of vape plus MHV has a further decrease in viability. Now, these mice groups are about 4 mice, so they're a little small. So we would need to look at bigger groups to see if there's an actual significant difference in this MHV versus co-exposure of vape and MHV. So with this, we concluded that vaping has shown to have negatively affect the morphology in these mice lungs or damage these mice lungs, and has also increased this inflammatory response in these lungs. And we also saw that vaping, when combined with coronavirus, showed this decrease in mice survivability. So for our future directions, we want to look at vaping toxicity and various cell lines and see if there's a difference in this inflammatory response between people in the population. Another avenue of our research investigated calcium signaling deregulation. So we want to further investigate what role calcium plays in how this disease affects the lungs. But overall, the goal of this project is to investigate the biological and social factors that increase COVID-19 risk. Do you have any questions?
We have a question from Kizzy. They asked, do you believe that different flavors are composed of different chemical components? Therefore, do they impact the body differently?
Okay, so that's another avenue that that's another future direction that our lab wants to look into. Because right now we use VapMate, VapMint to see what kind of damage it does to the lungs. But we want to look and see if because we know vape mints will common, and we've seen this damage that it causes. So we want to look and see if other flavors, if this mint flavor or other flavors lead to more severe versions. But it's one of the other avenues for our future studies.
And then Marianne asked if you can elaborate a nicotine level. Would that influence at all?
That's also another avenue that our future research is looking at right now, because our research first looked at just vaping and what role by itself. But now we want to look at if nicotine, aside from the chemical, will cause this lung damage. That's actually what we're doing now in lab.
And then Eduardo asked, what's the difference between a negative control and a vehicle control?
Let me I just want these diagrams to help me understand, explain. So with the vehicle control and the negative control, so the control, the PBS represents the negative control and the vehicle control, but the vehicle control is just used in our mechanism that we use to vape these mice to ensure there's no other chemical or what's the word I'm looking for? Pollution? What's the word I'm looking for? I'm sorry. I can't think of contamination. That's what I'm looking for. To make sure there's no contamination when compared to the vape mint in these lungs or in these samples. So the vehicle control, just to make sure that our mechanism that we're using is proceeding correctly. Mm-hmm.
And Jeffrey, I was wondering, with the treatment that vaped, were those mice exposed to coronavirus and then vaped? Or, because I imagine in human populations, like you're gonna vape, you're gonna smoke, then you're gonna get coronavirus, right? You don't get the coronavirus and be like, okay, I'm gonna take up vaping. At least I wouldn't think that would happen. So were those mice pre-exposed to vaping before getting coronavirus?
Yes, they were. Okay. They were exposed to vaping, and then some from our group were exposed to coronavirus.
Okay, thanks for clarifying.
Mm-hmm.
All right, that's all the questions I'm seeing for you right now, Jeffrey. So if you want to stop sharing your screen, we can kind of all come back together here. So I think there are a few outstanding questions for some of our previous presenters. Before I do that, I want to mention that you all did a phenomenal job. You got lots of good comments. For example, future Dr. Donia did a wonderful job raising awareness about mutations in minorities. Amazing works. I think you should all use that as your title, future doctors. That's awesome. So this question is for Asia. It came from Paula, and she had asked that question about the genes that you knock in. Where do those genes come from? Do they come from other zebrafish?
So like I said, transgenic fish, what you could do is you could have a gene from whatever you want. You're trying to get that gene into the fish genome. So that's how you could also do it. You could have it from possibly like a rat, a human, or something like that.
Thank you. And Jeffrey, another question just came in for you. How do you simulate vaping in mice?
So there is a mechanism we use that kind of heats up the chemical and then intranasally injects it into mice. So that's what we use to vape these mice, to simulate this vaping, is we inject this heated chemical, or we heat this chemical up, and I'm sorry. What's the word I'm looking for? Present it to the mouse airways through the nose to stimulate that vaping process. I mean, because obviously mice can't like vape out their mouth or whatever, so that's not.
All right, thanks, Jeffrey. Looking at the chat now, I don't see any more questions coming in. me give one final scroll through. I think that's it. Y'all did a fantastic job. Thank you for participating in SciTech Expo. For those of you watching at home, if you'd like more great programs like this, we have a whole week of activities and presentations coming up. So please check our schedule. You can visit the museum's website for all of our details. But thank you for attending today's program, and we hope to see you tomorrow. Great job, everybody.
Have a nice day, you guys.
Thanks. Presenters, can you stay on for 1 minute for me?